right, this is a little preview for our next lab. This is AP Lab number 2, Osmosis. So as you prepare to do this lab in your lab notebook, remember that you have a checklist on the inside of your notebook, and you need to make sure your pages are numbered through this lab before you turn this lab in, and you need to update your table of contents. So for table of contents, make sure you put the number of the lab, which is 2, the title of the lab, which is Osmosis, and the dates that we complete the lab. This is a 2-day lab, so we are doing this lab on Monday and Wednesday next week. All right, for your first page of your lab, you're going to repeat the title of the lab and the number, so you're going to put AP Lab number 2, Osmosis, and the dates completed. So then after that, you're going to write your background information. The lab does provide for you a little bit of an overview of the lab, so you can use that. And then remember, you can write more about water potential and hypertonic, hypotonic, diffusion, osmosis, those kind of things for this lab, because that, if you look at your vocabulary that is listed here, that is what this lab's about. All right, so your background information is talking about water potential, and we're going to be using 3 different kinds of potatoes for this lab. You are going to figure out the water potential of a regular Idaho potato, a sweet potato, and a rutabaga. And so we're going to figure out what the water potential is, which means which one has more water or which one is most juicy, if you think of it that way. In order to do that, we're going to put it in different solutions of sucrose, different molarities of sucrose, and then you're going to figure out what the molarity is of those potatoes in order to figure out the water potential for it. So that's what you're going to write for your background information. Your vocab is listed there. Notes will be helpful for that. A lot of those terms are from section 2 of your notes for chapter 5. All right, after that, you're going to do the purpose. There's only one part for this, so the purpose of this lab, I already told you, is to identify the concentration of the sucrose solutions, because you will not know which solution is 0, which solution is 0.2 molar, which solution is 0.4 or 0.6, etc. You will have no clue. They're going to be color-coded with letters. So that's our first goal, is to figure out the concentration of sucrose, and then our second goal is to figure out and determine the water potential of those 3 kinds of potatoes. Make sure you do your variables and your hypotheses. So you will need 2 hypotheses, one alternate, which is a guessing on which potato do you think will have the highest water potential or the lowest water potential, and then the null hypothesis, which basically says that they will all have the same water potential. Okay, so for your drawings, your setup, that's what you want to look at your procedure and how you're going to set this up. So looking at the procedure, it says to obtain 5 pieces of each potato. So you're going to have 5 rutabaga, 5 sweet potato, and 5 regular pieces of potato. You're going to have 5 cups, and you're going to label those cups A, B, C, D, E, and those A, B, C, D, E refers to the different colored solutions that you'll have to put in on top of those potatoes. So they'll be 0.2, 0.4, 0.6, 0.8, and a 0 molar solution, and there'll be different colors of food coloring. You will not know which one's which. Before you put them in the solution, you need to weigh them. So each group will have their own scale, and you're going to weigh these 5 pieces of potatoes individually and record that into your data table. So after your drawings, after you draw these 4 cups or 5 cups with the potato inside them, you're going to actually cut out your data tables. So I provided you data tables here. You're actually going to cut them out, and you can see this is the one that's going to figure out which molarity is which solution, and you can't do anything until you figure that out. And then you have a regular Idaho data table, a rutabaga, and a sweet potato is on the next page. So if you look at your data table, you need a title for each one, but if you look, it says solutions A, B, C, D, E, and you have an initial mass. So you need an initial mass of that potato before you put it into the cup. And then when we get the cup out of the refrigerator overnight, which will be a 2-dayer, you're going to get a final mass. So you will weigh it, dry it, and you will weigh it again, get your mass difference, and then percent change in mass. And then we're actually going to get class data and do a class average percent change in mass. Okay, so your picture just needs to show the 5 cups with the pieces of potato in there. We're only going to use 5 cups, which means each potato type will go into 1 cup. So 1 cup will have a piece of sweet potato, rutabaga, and Idaho in there, and we will cut them in different shapes so you will know which potato is which. Okay, after we do that, you will make a graph just like you did in the pre-lab to figure out your water potential. So there is no stats technically in this lab. It's water potential calculations for this lab. You will do a CER that I claim that this potato has the most water potential, and you would prove that with your calculations and do evidence and a reasoning. You will have an error analysis, what went wrong in the lab, and a further investigation.